In 1948, an Italian zoologist named Tina Franceschi took a handful of dried moss from a rooftop in Rome, a moss that had sat desiccated in the sun for 3 years without a drop of water, and she rehydrated it in a glass dish. Within hours, something in the moss began to move. Not bacteria, not spores, animals, eight-legged, round-bodied, lumbering creatures, each one smaller than a grain of sand, pulling themselves forward on stubby clawed feet, extending their round mouths to feed, resuming their lives with no apparent awareness that they had been suspended for 3 years in a state indistinguishable from death. We call them tardigrades. We also call them water bears and moss piglets. They have been on Earth for more than 500 million years. They have survived all 5 mass extinctions. They have been exposed to the vacuum of space, to radiation levels that would kill every other animal on Earth, to temperatures ranging from near about zero to well above boiling. They can be frozen, they can be crushed, they can be dried to a husk and revived decades later. They weigh less than a microgram, they are 0.5 millimeter long, and they are, without any reasonable competition, the most indestructible animal that has ever lived. We have known about tardigrades since 1773. We have had to and a half centuries to figure out where they came from, how they survive, what should kill them, and where they fit in the great tree of life. And for most of that time, we were wrong about nearly all of it. The questions seemed simple. The answers kept evading us. Then the genomes arrived, and what they revealed was not just strange. It was, by the standards of animal biology, almost incomprehensible. This is what the DNA actually shows. Before we get into it, hit that subscribe button. Every video here digs up the real origin of something you thought you knew. Let's begin. The old story of the tardigrade was not so much wrong as it was incomplete in ways that turned out to matter enormously. The textbook placed tardigrades in their own phylum, Tardigrada, small animals with 8 stubby legs, a through gut, a distinctive mouth apparatus called a bucopharyngeal apparatus used for piercing and sucking plant cells or prey, and an extraordinary ability to survive extreme conditions. Their closest known relatives, the book said, were the velvet worms, called onychophora, and further out, the arthropods. Together, these 3 groups, tardigrades, velvet worms, and arthropods, formed a supergroup called the ecdysozoa, the molting animals, named for the fact that all of them shed an external cuticle as they grow. The placement was reasonable, the anatomy made a certain kind of sense, and the age of the group was estimated, based on fossils, at somewhere around 500 million years, placing the origin of tardigrades at the great explosion of animal life in the Cambrian period, when nearly every major animal body plan appears in the fossil record within a geologically brief window. There was one problem the textbooks never fully addressed. The survival. Tardigrades can survive things that should be physically impossible for any animal to survive. The mechanism they use is called cryptobiosis, a state in which all measurable metabolic activity stops. The animal is not sleeping, it is not hibernating, it is not in any state that biology had previously described as life. It is a dehydrated husk, its internal water content reduced to less than 3% of normal, its body folded into a compact barrel shape called a ton. In this state, it produces no carbon dioxide, shows no electrical activity, responds to no stimulus. By every conventional measure, it is dead. And then water comes. And it is not. For most of the 20th century, the mechanism behind this survival was attributed to one compound, trehalose, a sugar that many organisms produce under desiccation stress, which forms a glassy matrix around cellular structures and proteins, replacing the water molecules that normally keep them stable, protecting membranes and molecular machinery from collapsing while the water is gone. The tardigrade textbook story was that tardigrades made a lot of trehalose, and that was how they survived. Simple, satisfying, wrong, or rather, not wrong exactly, but catastrophically incomplete. because when the first complete tardigrade genome was sequenced in 2015 by a team at the University of North Carolina led by Thomas Boothby, the Trehalose story was only the beginning of what the data revealed, and the first thing the data revealed was a scandal. The initial analysis of the tardigrade genome produced a finding so extraordinary that it made international headlines. The genome of Rhamnotsodius variornidus, one of the hardiest tardigrade species, appeared to contain an extraordinary amount of foreign DNA, not tardigrade DNA, DNA from other organisms, bacteria, fungi, plants, archaea. Genes that had no business being in an animal genome were sitting inside the tardigrade genome as if they had always belonged there. The initial estimate was that roughly 17% of the tardigrade's genes had been acquired from non-animal sources through a process called horizontal gene transfer, the direct incorporation of genetic material from one organism into another without sexual reproduction. This number was shocking. In most animals, horizontal gene transfer is vanishingly rare. In the tardigrade, it appeared to be a dominant feature of the genome. The finding was immediately controversial. Other researchers argued that the foreign DNA was contamination. Bacteria and fungi that had been living on the tardigrade samples and whose DNA had been inadvertently sequenced alongside the tardigrade's own genome. The scientific argument that followed was sharp and public. The final answer, which emerged from multiple independent genome sequencing efforts across multiple species over the following years, was more nuanced than either extreme. The tardigrade does engage in horizontal gene transfer at a rate that is genuinely elevated compared to most animals. The mechanism appears to be related to the extreme desiccation and radiation damage the animal routinely survives. When a tardigrade is dried to a ton and rehydrated, its DNA is fragmented by the physical stress of the process. The cellular machinery that repairs this damage appears, in some cases, to incorporate DNA from the tardigrade's environment into the repair process. The tardigrade is, in effect, using the stress that should kill it as an opportunity to absorb genetic material from the organisms around it. This is not contamination. This is the tardigrade genome behaving as a living archive of its environment. Every time a tardigrade survives something that should have destroyed it, it may be returning to active life with a slightly different genome than it had before. But the horizontal gene transfer story, dramatic as it was not the deepest revelation in the genome data. The deepest revelation concerned the proteins. When Boothby and his team looked at what the tardigrade genome actually produces, they found a class of proteins that had never been seen before in any organism. The team named them tardigrade-specific intrinsically disordered proteins, or TDPs. These proteins, when expressed in a desiccating cell, do something physically remarkable. They form a gel, not a liquid, not a solid, but a biological glass, a vitrified matrix that encases the cell's contents and holds them in place while the water disappears. This is not the same as what Trehalose does. Trehalose works at the molecular level, surrounding individual proteins and membranes. The TDPs work at a higher organizational level, vitrifying the entire cellular interior, encasing the cell's machinery in a protective glass that prevents the collapse and aggregation of proteins that would otherwise destroy the cell's function permanently. When the researchers took the genes encoding these TDPs and inserted them into yeast cells that are not tardigrades, not even remotely related to tardigrades, the yeast became more tolerant of desiccation. The tardigrade's survival mechanism was portable. It could be borrowed, inserted into a completely foreign organism, and made to work. This finding had implications that extended well beyond the biology laboratory, because the TDP proteins do not just protect cells during desiccation. They protect against radiation damage. They protect against pressure. They protect against temperature extremes. They appear to be a general-purpose cellular protection mechanism, assembled by the tardigrade genome through an evolutionary process that is still not fully understood, that shields the cell from a remarkably broad range of physical insults. The question of where these proteins came from, how they evolved, and why no other animal lineage appears to have developed anything quite like them is one of the deepest open questions in the genomic study of tardigrades. The third revelation from the genome was the one that rewrote the family tree. For decades, the position of tardigrades within the animal kingdom had been debated. Were they closer to arthropods, to roundworms? Were they a truly ancient group that had branched off before the great divergences of the Cambrian? The physical anatomy gave ambiguous signals. The legs were suggestive of arthropod ancestry. The molting was consistent with ecdysozoa, but the precise placement kept shifting with every new analysis. The genome settled it, but in a way that nobody expected. Multiple large-scale phylogenomic studies, analyzing hundreds to thousands of genes simultaneously across all major animal groups, converged on a placement for tardigrades that was surprising. Tardigrades are not, as some earlier analyses had suggested, a basal or primitive lineage sitting near the base of the animal family tree. They are nested within the ecdysozoa, most closely related to the roundworms, the nematodes, specifically, and the arthropods come next. But the more significant finding was the age. The molecular clock analysis, calibrated against known fossil dates for other animal groups, pushed the origin of tardigrades much deeper than Cambrian fossils had suggested. The divergence of the tardigrade lineage from its closest relatives appears to have happened in the late Precambrian, potentially more than 600 million years ago, before the Cambrian explosion itself. This means the tardigrade body plan, that stubby eight-legged architecture, the bucopharyngeal apparatus, the basic life history that allows cryptobiosis, was established before most animals with eyes, legs, or complex nervous systems had appeared on Earth. The tardigrade watched the Cambrian explosion happen from the sidelines. It was already old. And then it survived what the Cambrian world produced, and what the Ordovician extinction took, and what the Great Permian Triassic extinction erased, and the End Cretaceous impact, and everything else. 5 extinctions, each one killing the majority of complex life on Earth. The tardigrade, in its ton, suspended, dried, waiting, outlasted them all. But here is the part that the genomic story makes most unsettling, because the survival is not just ancient. The genomic data reveals that it is still being refined. When researchers compared the genomes of multiple tardigrade species, ranging from the extremely desiccation tolerant to the moderately tolerant, they found that the TDP protein families and the DNA repair gene families show signatures of ongoing positive selection, meaning evolution is still actively working on the survival machinery. The proteins are not a finished solution, they are a work in progress. The tardigrade is still, right now, getting better at not dying, and the fourth revelation from the genome is the one that holds the darkest implication for the rest of us. Tardigrades can survive radiation doses of 570,000 roentgens. For context, a dose of 500 roentgens is lethal to a human. The tardigrade can absorb more than 1,000 times what would kill us and resume life as if nothing happened. The mechanism, revealed in the genome, involves a protein called SUP, damage suppressor. SUP physically coats the tardigrade's DNA, binding to the chromatin and shielding the double helix from the ionizing radiation that would otherwise shear it into fragments. When researchers inserted the SUP gene into human cultured cells, the human cells became approximately 40% more resistant to radiation damage. A protein from a half-millimeter water bear, evolved over 600 million years of radiation exposure, offers partial protection to human cells. The tardigrade has been solving problems that we have not yet invented solutions to, and its genome is the record of those solutions, accumulated across more than 500 million years of surviving what should not be survivable. There is one final layer to this story, and it concerns what happens to tardigrades when we send them somewhere no animal should ever go. In 2007, the European Space Agency sent 3,000 tardigrades into low Earth orbit aboard the PHOTONM-3 satellite. The tardigrades were exposed to the full vacuum of space, ultraviolet radiation from unfiltered sunlight, and cosmic rays for 12 days. When they were brought back and rehydrated, 68% of them were alive. Some had reproduced in space. Their eggs hatched normally. In 2019, an Israeli spacecraft called Bereshit crashed on the moon. It was carrying a payload of dehydrated tardigrades. Nobody knows if any survived the impact. The moon has not been revisited to check. There may be, right now, tardigrades on the lunar surface, not alive in any active sense, in ton form, waiting, as they have waited through 5 extinctions and 600 million years. If the moon is ever visited again, and those samples are recovered and rehydrated, there is a reasonable scientific possibility that they would resume their lives. The organism that survived the Permian extinction, the Ordovician extinction, the end Cretaceous impact, and a decade of laboratory radiation experiments may have outlasted the first spacecraft humanity ever crashed into another world. We spent 2.5 centuries studying tardigrades before we read their genomes. We thought we understood them. We thought they were interesting but simple, extreme survivors with a sugar-based trick and a peculiar body plan. The genome revealed something else entirely, an animal that collects foreign DNA from its environment as it heals, an animal that carries glass-forming proteins found in no other creature on Earth, an animal whose radiation shield can be borrowed by human cells, an animal whose survival mechanisms are still being refined by evolution today, after 600 million years, because the problems are not yet fully solved. The tardigrade is not a relic. It is not simple. It is not merely extreme. It is, at the genomic level, one of the most sophisticated biological systems ever assembled by evolution, built across more than 500 million years of solving problems that nothing else survived long enough to solve. In 1948, on a Roman rooftop, something that had been dry for 3 years began to move when water returned. We thought we knew what that meant. We thought it was a curiosity, a trick of chemistry. The genome tells us it was something else. The end point of a 600 million year experiment in making life impossible to end. The DNA is only beginning to tell us what it knows. If you found this story remarkable, consider subscribing. There are more origins to uncover, and the DNA is only beginning to tell us what it knows.